but now we have to be inside for another 50 minutes. Well, unless we can find a way to do projection outside, in which case we could do class outside sometimes. That would be nice. Before we get started, if you're willing to, I would love to get feedback on the class at any point, but I woke up early this morning and I got this kick to build this survey. So I built a survey that's an anonymous survey that's no points. I'm just wondering if you will give me feedback on a few aspects about course load, that is assignment load, and your study habits in the class. So anonymous survey, if you want to take it, I'd really appreciate your feedback. No points. It's anonymous, which is why it's no points, because if it's anonymous, then I have no way to give people points. And as you just heard, apparently there's an MCAT class tomorrow where you can get Chick-fil-A. You heard about the person that got stabbed over the Chick-fil-A sandwich in, where was it? Or it was Popeyes, never mind. Yeah, no one's stabbing anywhere but Chick-fil-A? Whatever. Okay. To start, do please go on to Qualtrics if you have time and take this survey. I'd appreciate your feedback. Last. So, did you know that chicken is made up of amino acids? Speaking of Chick-fil-A and Popeyes. So when we closed class last time, just starting to talk, and we haven't hit translation yet. We're just starting to talk about transcription, but this is a good point to start talking about amino acids as well. So at the end of last class, I had started talking about amino acids. Some of this may be familiar from organic chemistry or chemistry, previous classes, or it might not. The simple rule that we talked about last time is that these condensation reactions between the carboxylic acid group of one amino acid, each of these different amino acids has an R group, which is a generic code for whatever is sticking off of the central carbon on these atoms. So the R groups I'm circling in black, just the top row. So this is how the amino acids differ. So when you have a condensation reaction between the amino group of one amino acid with another R group, that takes two H's and one oxygen and builds what we talked about last time is this bond. Which is attached to carbons with the other R groups of the two amino acids on either side. This gives us both polarity, that every amino acid has an amino terminus, like here in phenylalanine in red, there's an amino terminus, or an N end, and a C end, or a carboxylic acid end. And that's the polarity that proteins have. One end has a hydrogen and nitrogen, the amino group, and the other end has a carboxylic acid group. And this bond, lastly, for review, is called what? It's a peptide bond. I know why it's called a bond. I don't know why it's called a peptide bond, but it links two peptides together, so maybe that's why it's a peptide bond. All right, so today in class, we've already covered most of the vocabulary for the genes. We're going to move into transcription now. Does anybody have any questions about any of the vocabulary items or tasks? We've done most of them, at least. The second task here was based on that SRY case study paper from last class, looking at the agarose gel and evaluating the experimental results based on control experiments. That's what I mean by that task specifically. We'd already done polymerase gel electrophoresis. We did that last topic. And we've done quite a bit of NCBI and BLAST, or at least a little bit, and we're just going to move into amino acids and proteins. Yeah?
Can you briefly go over the necessity and sufficiency thing again?
Absolutely. So the very last bit, genetic necessity and genetic sufficiency. So there are different types of analyses that we use to identify whether a gene is necessary to do something, a gene is necessary to have an effect, and if it's sufficient to have a particular effect on an organism. So necessity and sufficiency. So if a gene is necessary to do something, that means you have to have it. For example, the example we've been using, SRY is necessary to produce males. If you don't have SRY, you can't become male. So what is the sufficiency argument? Different than necessity. There's have to have it versus if it's enough, if that's all you need. Which again is, SRY is from an XY mouse, we saw moving that into an XX mouse. And SRY was sufficient. It was all that was needed to move, the only gene, the only genetic material that was needed to be moved from a male mouse into an XX mouse to cause that XX mouse to become male. So if you are a male, you need it. It's necessary for male sex determination, and it's also the only thing you have to move into an organism that doesn't have it to cause that organism to become male in this case. Yeah.
So if I understand correctly, necessity, if it's necessary, then it's sufficient, but it doesn't necessarily mean that if it's sufficient, it's necessary.
Right. So if it's, I hate to make a hasty logical leap. I think what you just said is accurate. So let me restate, and then I'll have to think about it more to make sure that I'm not misremembering any other weird cases in which that wouldn't be true. So is it, what you're asking is, is it the case that if it's necessary, then if it's sufficient, then it must be necessary? Right? What's the relationship between necessity and sufficiency is what you're asking. Is that right? Yeah. So if it's necessary to do something, then it's not necessarily all you need. So for example, there's a gene that SRY, and we haven't talked about this in class, one of the genes that SRY turns on is SOX9. So if you have SRY, that transcription factor, a protein that causes other genes to be transcribed, goes around and turns on a bunch of other genes like SOX9. You also have to have SOX9 to become male. So it's necessary for being a male, because it's a gene that turns on other genes that create male traits. But if you moved SOX9 into a female, an XX organism, it stays female because SRY isn't there to turn on SOX9 to turn on the rest of the male determining genes. So that's a case where there's a gene that's necessary for male development, but by itself, just that one gene can't turn on male sexual development. So if you move it, it's not sufficient, in other words. It's necessary but not sufficient. If it's sufficient, then I agree with you. I think it has to be necessary. But this is the logic I'm going to have to think about. I'll report back next time. So this is the question. If a gene is sufficient, is it always undoubtedly necessary for the process? So I will report back. Thanks for the question. Good to clarify. So we just looked at the case where a gene that's necessary for something isn't sufficient. I'm going to see if I can come up with an example of the opposite. Is there any case where there's a gene that's sufficient but not necessary? Doesn't seem logical that that would exist, but I need to look into it. Other questions? So, transcription. And we're going to start with transcription initiation. This is getting at this question in the bottom right from the class trailer video. How does a cell know where its genes are? How does a cell know where in the DNA genes are located? Which parts of DNA RNA polymerase should transcribe and make a messenger RNA. And the next topic after this one, after transcription initiation, is going to be processing. So what happens to the messenger RNA is that's going to be splicing, capping, and polyadenylation, terms you may or may not remember. And then after that, we're going to talk about the last step, translation and the production of proteins. So most of the vocabulary up here, all of the vocabulary up here is focused on that one point of transcription initiation. How does messenger RNA production start? How is it initiated? Where does the cell know its genes are? How does the cell know? So one of the case study, or the case study, has to do with this three-dimensional protein structure. Was anybody able to effectively open this and look at it and play with it? Did anybody do it? So a few. I'm going to pause the recording then and show you what it looks like on.
Can you relate this to the positive
This, so TATA binding protein doesn't directly relate to histones, but it does only interact with DNA in open chromatin structure, so in parts of DNA that are not tightly associated with histones, which we haven't talked a lot about chromatin structure in class, so don't worry about that aspect if you don't want to. Okay, so here's another picture from the case study itself in the course manual. Now the red is the double helix. We're looking down the double helix, and here's TATA binding protein. How would you describe the interaction? We just talked about that, and what this has to do with transcription. So this is TATA binding protein. The molecule in red is DNA. The other bit is TATA binding protein. And they're binding to each other chemically. And what this has to do with transcription is TATA binding protein, when it does this to DNA, when it finds the DNA sequence it's specific for, it will interact with DNA like this. And then, this is all review from the video. What does TATA binding protein binding DNA do for transcription? So that protein, TBP, TATA binding protein, binds to DNA, and then what happens? It has two functions. One surface of that protein is a DNA binding domain. That is, it's a structure of polypeptides. It's a protein structure that feels for that particular DNA sequence and will stick to it. What's the other thing that TATA binding protein does? It's got another domain here. Another function, in other words. It doesn't just bind DNA. It interacts with another protein. What's the other protein? RNA polymerase. Which I'll abbreviate RNAP. That's the protein that does transcription. Reads DNA and produces an RNA transcript. The reason that TATA binding protein is critical is because RNA polymerase itself doesn't have a DNA binding domain. That is, RNA polymerase, that protein, doesn't know where the genes are. It doesn't have a surface, a domain, that recognizes this particular DNA sequence that TATA binding protein does. So maybe an analogy for this is TATA binding protein is kind of like the GPS for RNA polymerase. TATA binding protein tells RNA polymerase where to go. The first step is TATA binding protein binds to DNA. The second step is, because TATA binding protein and RNA polymerase can stick together at this surface where TATA binding protein binds to RNA polymerase, TATA binding protein grabs RNA polymerase and holds it nearby the promoter, nearby where the transcription is going to start. So where in this figure is the transcription start site? Where's the first nucleotide that RNA polymerase is going to read and produce a messenger, start producing a messenger RNA? Is it over here?
Yes.
Is it over here? It's somewhere where RNA polymerase is being held by TBP near the DNA. So it's somewhere in here. Right, so that's what TATA binding protein does. TATA binding protein binds DNA, grabs RNA polymerase, says, get over here, holds it near the DNA until other proteins can unwind the DNA and RNA polymerase starts reading this one strand of DNA and producing RNA. So that transcription start site is somewhere in here, and we usually indicate a transcription start site with a right-handed, right-angled arrow pointing in the direction that RNA polymerase will move to read the gene. And what number do we assign the transcription start site? It's a new form of DNA sequence numbering. Not polarity, still 5' and 3'. First nucleotide in a transcript is plus one. It's called the +1 nucleotide. The first nucleotide that RNA polymerase reads the DNA version of and produces an RNA molecule is the +1 nucleotide. And because RNA polymerase then moves to the right, and we only know that because of the way that I've drawn this arrow, at least for now, that's the only way we know it, then which nucleotide is the next one, numbering-wise? +2, and then the only other thing we need to review is, if we move in the opposite direction, what numbers of nucleotides are TATA binding protein binding? Negative. Yeah, it's -10 to 30, -20. It's about 20 nucleotides upstream of the transcription start site. You don't need to know a specific number, but it's close by. It's a few nucleotides to the left, and those are numbered negatively. So the first nucleotide to the left of +1 is -1. You keep counting negatively as you move to the left. So this is the answer to how does a cell know where to start producing transcripts. It has almost, at least the most important thing for you to learn in this class, is it has entirely to do with where TATA binding protein binds DNA. That's the first step. If TBP binds to the DNA, then it will recruit RNA polymerase, hold it there until RNA polymerase starts transcribing.
Can you point out where the promoter site is?
Okay, great question. So there are two different, and we'll review this several times today or today in next class, depending on how quickly we move. So where in this figure is the gene in the DNA molecule? The gene is,
One down to wherever it is.
the gene is, at least starts at the left side at +1. And then we'll define where the right end of the gene is at some point in the future. That's the next topic, how RNA polymerase knows where to stop transcribing the gene. By contrast, then everything to the left for a little while is what we call the promoter. We generally classify genetics or genetic molecules, DNA, into 3 different types, of which 2 are up here. Promoters, which are DNA sequences that regulate gene expression, whether or not transcription happens or not. That includes TATA binding protein's sequence that it identifies. It's in the promoter. Promoter is not a part of the chromosome that gets transcribed. That's the gene. So promoter, DNA that regulates when the gene is turned on or how the gene is turned on, and the gene itself is the thing that encodes an RNA molecule. So the promoter is regulatory. The gene is functional. Whether or not it produces an RNA that's just meant to be an RNA or an RNA that gets translated into protein, that's the part that does the work of the cell, the gene. The promoter contains DNA sequences that control the gene. Other questions? General principle of transcription is also on the header, so I would be remiss if I did not address that at least briefly. TBP recognizes specific DNA sequence, which we're going to talk about for the rest of class, at least. It's going to be really boring. We're talking about one DNA sequence for the whole rest of the class. But before we get to the boring part, what happens to TATA binding protein's ability to bind DNA if that DNA sequence it recognizes, whatever it is, gets mutated, changes? Right. Say it louder.
You can't read it?
TATA binding protein recognizes a particular DNA sequence. If a mutation happens and that sequence changes, then TATA binding protein doesn't bind as well, or as long, or as tightly, or at all. So then what happens to transcription of this gene?
It's a little messed up.
It gets more transcription or less transcription?
Less transcription.
Less. So this is the really important principle of transcription, is that the genes that turn on transcription, like TATA binding protein, have a particular DNA sequence they bind best. There are other DNA sequences that they could bind okay, or poorly, or not at all. And mutations then can change how tightly and for how long, in terms of time, TATA binding protein will stick to a particular piece of DNA. And the more frequently TATA binding protein binds the promoter, the more frequently RNA polymerase gets brought to the promoter and transcribes the gene. In other words, increased TBP binding correlates with, wow, that's horrible penmanship, correlates with an increase or a decrease in transcription. So if TBP binds better to the promoter, you get more transcription. Increase. Not a decrease. So the direct relationship between how well TBP binds the DNA sequence it recognizes and how much transcription occurs. Positive correlation. So what is the DNA sequence that TBP recognizes? The actual sequence.
TATA?
It's called TATA binding protein because it recognizes the sequence TATA. And then a couple other A's. So depending on who you ask, there's a different ideal or consensus sequence. We've seen consensus sequences before, and that's why we worked on that a lot at the start of the semester. So this protein loves to bind TATA. It seems to be like the ideal DNA sequence it binds. So what we're going to look at now is how scientists discovered that that was the sequence that TBP likes best. I won't take too much time on this. Maybe in future semesters I'll turn this into a case study for some lucky class. So here's how the scientists did this project. So briefly, this is an approach we use all the time in the lab called reporter gene assays. It's a great way to understand how well transcription is occurring in any sort of a system. So to have transcription, you have to have, in this case, the TATA sequence. That's where TATA binding protein will bind. And we've already said that that region of the gene is the promoter. And then to the right of the start site is what? LacZ. It's a type of, what are we going to fill in here? Promoters on one side.
Gene.
This is the gene. So LacZ is the name of a gene in yeast. Horrible gene name. I was not involved in the naming of the genes, so I apologize. But that gene encodes the protein beta-galactosidase. So the gene is called LacZ. The protein it produces after transcription and translation is beta-gal, for short, beta-galactosidase. Oh, and let's see, the transcription start site, that's the +1 nucleotide. Great. So, in this case, if TBP is present, will we expect to see transcription of beta-galactosidase? In other words, will TBP recognize that sequence in the promoter? Yes, and we'll bind to it. TBP recruits RNA polymerase, and then transcription and translation, TXN, TLN, transcription, translation, you get beta-gal. So the protein gets produced. Pretty straightforward. You've got a TATA sequence, TBP binds it, RNA polymerase is brought in, the gene, whatever the gene is, in this case beta-gal, or LacZ rather, gene, is transcribed and translated, and beta-galactosidase is produced. So simple system that scientists use in all sorts of organisms, bacteria, yeast, cells that are from humans that we grow in tissue culture. What happens now if we fiddle with the TATA? We already discussed this, just asking again. What happens if we mutate that 6 nucleotide sequence?
Protein's not gonna, can't transcribe it.
Not as well.
Right, so there's going to be a continuum. There might be some mutations that don't affect TBP binding. There could be some mutations that totally abolish it, do away with TBP binding. So the effect is hard to know until you do this assay. So here's how the beta-galactosidase assay works. Has anybody done this, by the way? Lab classes, some of you may be in research groups that use beta-gal assays. If not, you probably will, especially if you take microbiology at any point. You may wind up doing this. I'm not sure if you do. So beta-galactosidase, here's that enzyme. It's involved in this chemical reaction. It takes this compound called XGAL and removes the sugar molecule from that. So it's called beta-galactosidase. It aces the galactose, excuse me. And that releases this bit, and 2 of those compounds will dimerize and form that compound on right, which I don't remember its IUPAC name. The important thing is that XGAL is clear, it's colorless, but this a molecule that's on the right is a blue color. So if you have beta-galactosidase and you feed some organism, you throw some XGAL on an organism, if beta-galactosidase is present, then the organism turns blue, because beta-galactosidase is reacting with this chemical to produce a blue product. It's a blue dye. So if TBP can bind the DNA, beta-gal is produced. And if beta-gal is produced, then the organism turns blue. So this is our assay for, is TBP binding DNA? If it does, it produces beta-galactosidase. Beta-galactosidase does this chemical reaction, and you see what's happening by blue. Blue means transcription. Blue means our DNA was being transcribed into RNA by TBP binding. So here's your chance. We've got 3 different experiments in silico on computer, thought experiments. These are identical conditions, except for one thing has changed. There's a TATA binding site, there's the transcription start site for LacZ, and in the presence of XGAL, how blue will that condition be? A lot blue, a little blue, no blue. The only difference between 1, 2, and 3 is the DNA sequence of that same TBP binding site. So I'd like you, please, to make predictions. How blue will conditions 1, 2, and 3 be? You can use your scale of blueness if you want. What happens in number 1?
A lot.
Yeah.
That was the case we talked about already. Normal TATA binding sequence, TATA. So how much blue?
Same as the control.
Blue, blue? Blue, blue.
Same as the control.
Super blue. We're going to call that plus, plus. Lots of blue. Just for now, plus, plus. That is not genetic terminology. That's just something that I made up. And this is a control experiment. Which type of control experiment is this? Positive or negative?
Negative.
Hard to know. We haven't really discussed it a lot. The positive control in an experiment is the one you know should work or shouldn't work. Should work. Should work. Positive. Good. Yes. So this is kind of a positive control experiment because we haven't done anything to the system. We expect that if anything is going to produce beta-galactosidase and make lots of blue pigment, it was going to be this condition, where TBP's binding site is the best one that we know of. So then what happens in experiment 2? Is there going to be blue?
Yes.
I like your confidence. Possibly. We don't know. That's why this is the experiment. It turns out the answer is yes. TBP can bind that sequence. It just binds it less well. So it brings RNA polymerase to the promoter less frequently, so less transcription occurs, so less beta-galactosidase gets produced, this protein, and there's less protein, so it produces, in the same period of time, less blue pigment. Fewer molecules of enzyme means fewer molecules of dye get produced after this reaction. So maybe that's just blue, little blue, 1 plus. And then what happens if you get rid of the TATA binding sequence altogether?
No binding would happen.
You would expect,
Little to no binding.
yeah, that's the negative control. The case where you expect nothing to happen, where you expect your experiment not to produce anything. So if there's nothing for TBP to bind, you would expect no transcription, no beta-galactosidase, no blue. Now this is all an ideal thought experiment situation. This is mostly how a beta, and this is just something I stole off the internet without attribution, so I apologize to the owner of this. I forgot to note where on the internet I found this. But it's an example of this type of assay. You could consider, for example, that strain 3 maybe has a TA sequence, so those are the bluest. TBP, best match to its binding sequence, most transcription of beta-galactosidase, most production of this pigment. Which one is the one where we deleted, in theory, on this picture? Which one would be representative of the one where we deleted the TATA binding protein sequence altogether?
4, 5, and 6.
4.
Yeah, one of these ones, or most of them. So dash, 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 no TA at all, no beta-galactosidase produced, no blue pigment, and then maybe TGTA looks something like columns 1 or 2. You've got yeast that are making some beta-galactosidase, and they're kind of turning blue, but not as well as the wild type. The positive control, in this case, I don't know that this is actually what these people were doing in this experiment. It's almost certainly not. But for our purposes, positive control, negative control, and the experimental condition where we don't know what the answer is until we do the experiment and see the yeast. Yeah.
In a previous class, we went over an experimental schema with bacterial DNA and xgal, in the presence of xgal. And it was basically putting the bacterial DNA on an antibiotic agar sheet and adding xgal and adding also some sort of splicing or some sort of gene to...
IPTG?
I think so, for the circular DNA to uptake.
Yeah.
And basically, if it had uptaken the gene, it would have been white in the presence of xgal, and
Right.
if it didn't uptake the gene, it would be blue in xgal. So I'm sort of confused in relation to it.
So there's, what we're talking about is there's a different type of experiment that isn't this one that uses the same system that reports whether or not transcription is happening in the presence or absence of beta-galactosidase. So since we haven't talked about that in this class, I will happily describe that to you after class if you want. But it's not, that also is assaying transcription, but not the way I just described.
It is the opposite, right?
So you still get, let's talk after class. Just so that I don't accidentally confuse everybody else by answering your question. Since that was not a previous genetics class, right? That was a previous other class, non-biology 102 you're talking about?
Yeah.
When you said previous class? Okay. I just wanted to make sure I hadn't brought up XGAL before because I thought I hadn't. So happy to talk to you about it. So here's what actually happened. And what I'd like you to do, we won't do this in class, but it is available for you now on Google Classroom. This is just to demonstrate what scientists did when they figured out that TATA was TBP's favorite DNA sequence. It's hard to see up here, but there are a bunch of nucleotides, so please, if you want to, go on Google Classroom, open this Google Sheet, and there are a bunch of cells that are colored in gray down here. What the scientists did was they figured out where transcription start was for a bunch of different unrelated genes in yeast. So these are all the +1 nucleotides of different imaginary genes. So they got DNA sequence, they knew where the transcription start site was, so they lined up, this is what you should do, please. Go do this multiple sequence alignment where you line up all of the transcription start sites, so it's just one column of gray nucleotides from top to bottom. So you're looking at all of the gene sequences relative to where they all start transcribing. And if you get them all lined up like that, then where would you expect to see a DNA binding sequence for a protein that turns on transcription relative to where transcription starts? Somewhere to the left, this is the promoter region. And then what you're looking for is a consensus sequence. And this is exactly what the yeast geneticists did. Once they lined up all these different genes where they knew transcription started, they had this idea that if that's where RNA polymerase is located, and if it's being brought to that +1 transcription start site by a transcription factor, of which TBP is a type. So a transcription factor is a general term for any protein that binds to a DNA sequence and turns on transcription. TATA binding protein is a transcription factor. It's not the only transcription factor. So they figured if they lined up all these DNA sequences at the start site, then maybe a consensus sequence would emerge somewhere in the promoter that would be whatever the sequence is that whatever protein it is does this job binds to. So I'll let you do that discovery if you want to. This is exactly what those scientists did a couple decades ago now, I think, at least, to figure out which DNA sequence TATA binding protein binds. And you could do this with, note to self, you could do this for any sort of transcription factor with any sort of DNA sequence. This is one of the reasons making multiple sequence alignments is important to geneticists. It reveals consensus sequences, and you might be able to figure out the consensus sequence for any particular transcription factor that's not TBP, hint, hint, if you do this sort of an analysis. We've done it before. Multiple sequence alignment, look for a consensus sequence. Any questions? So I'd like to end with a quick story then. And then everybody gets milk and cookies and goes to sleep. Not that kind of story. This is a story about TATA binding protein and the sequence it recognizes, and there will be a surprise at the end, and I hope you're surprised. I was. So that was all just made up data. If you want to go play on Google Sheets, I made this exercise for you to practice doing another multiple sequence alignment and to replicate what scientists do when they actually get the real DNA sequences. This is not the study that I was just mentioning to you where the scientists first discovered that TATA was TATA binding protein's favorite DNA sequence. So for example, here at the top of the screen, let me zoom in. What these scientists were doing in this study were they were trying to figure out how badly can we mess up TATA and still get transcription. So they were doing mutagenesis. They wanted to figure out what sorts of DNA sequences can you put in place of TATA and still get transcription. So for example, here is a DNA sequence that they refer to as DNA sequence 3801 that does have TATA in it. That should work pretty well for producing transcription, and it does. So the phenotype here, like the scheme we were just using, is ++. This construct in an XGAL assay, a beta-gal assay, produced a lot of blue. So they scored that construct as ++. Organisms that had this sequence in their promoter made lots of beta-gal, made lots of blue dye. So down below here are a bunch of different constructs that they replaced the TATA sequence with, and their outcomes. So what's expected and what's surprising about this? Do you see any of these below the dotted line that have the consensus sequence? TA, TA, AA. So where's one? 3824, I heard. So that's this one. So it's got TA, TA, AA in it. And what's its phenotype? How much blue does it produce? 3 pluses. Lots of blue. Okay. And so there was a pattern that they discovered that most of these, that somewhere in there had the consensus sequence, made a lot or some blue dye. X-gal was cleaved into this product that gives this blue coloration. What's weird about this? Surprise! What's odd?
There's 3835 that is TATAAA, and it only produces 1 plus.
Where's this one?
3835.
Oh, down at the bottom. 3835 has TATAAA,
And it's a 1 plus.
but it doesn't produce much beta-gal. In the other direction, what about 3821? 3 plus. No TATAAA. The take-home message is that it's more complicated than it seems, and it always is in genetics. That yes, TATAAA is preferred by TBP. It's sufficient for transcription. That is, if you have it most of the time, except for this exception down at the bottom and a few exceptions in this table, most of the time that sequence is enough to get transcription. Is it necessary for transcription? No, because we've got transcription going on up here in its absence. Lots of transcription, lots of blue color. So beta-galactosidase X-gal, the gene, is being produced, translated into beta-gal, cleaving X-gal, making blue dye, no TATA.
Is that our example of sufficient but not necessary?
Sure, let's go with that. Sufficient but not necessary. Yes. Thank you. Again, I will reflect on that to make sure I'm not making some wild claim to fame, but sufficient. I'll write this down for now. Sufficient but not necessary. So TATA. For transcription, sufficient but not necessary. So for next class, please take a look at this new question, one orphan gene. Orphan gene just means a gene that people think is only found in one species. Is it only found in humans and chimpanzees? Due Sunday night. Remember, we don't have class on Monday, so Sunday night's kind of an arbitrary deadline. And for next class, we're going to move into finishing up transcription. So the 3 exercises from the course manual, and then the 2 first attempt exercises for the next topic. And finally, if you would, again, for those of you that weren't